bilayer to me. Looks like a protein shaped in a channel. It looks like sodium is rushing in. Remember what that's called? Depolarization. It looks like potassium is rushing out. Repolarization. So that's creating a spark. The same spark that we talked about with the sliding filament theory. The spark that's used to create that electrical current that travels down into the T-tubules and opens up the cisterns. Oh my gosh, here we go again. But this time it's all about nerves. It's saying sodium rushes in, potassium moves out, and it creates a spark. So this spark is really important because this spark could be the spark that opens up the neurotransmitter floodgates. Remember, a nerve signal travels down the knob. That's the spark that we're creating here. So we're going to talk all about this spark and how it creates an impressive signal for the last 10 minutes of class, and then we'll continue with this on Thursday. So let's take a look at this word, and I want you to write down these two words, and there's a good chance that this will be a question on the exam. Literally, I promise you this. Is to be able to distinguish two types of sparks. One's called a local potential, and the other one is called an action potential. So can you distinguish a local from an action potential? Here's the third grade explanation. Local potentials are smaller and weaker. Action potentials are larger and faster. Okay, so here are some college explanations between the two, between a local potential and an action potential. It says local potentials are graded, which means they can get stronger and weaker. Action potentials, no. It's just one impressive burst of electrical energy. It says that a local potential becomes weaker as they get farther away from the cell body. So it says that they become decremental, which means weaker. Look at this. This is something that an action potential would never be. A local potential can be reversible. Go backwards. And it says, here's the big part, that it can be either excitatory or inhibitory. So let's get back now to the drawing over here on the right. And let's make sense of this. So I'm going to draw local potentials as being little tiny arrows. So here are some arrows, local potentials coming from some synapses from one neuron to another. And all of these little arrows kind of reach the bottom. And this area right here that I'm drawing in dots is called the trigger zone. So I describe the trigger zone as a place where the cell body merges and becomes the axon. But now we can say it's really the place where all of the local potentials accumulate. Remember, they can be excitatory or they can be inhibitory. So at the trigger zone, here we go, it's like a democracy. Majority wins. If there are more excitatory neurotransmitters or excitatory responses, then it's going to send an action potential. If there's more inhibitory, then it's not going to send. So your neurons are always getting blasted with a lot of different stimuli. I always think about, you guys sitting in this room. Most of you are trying to focus in on what you're seeing and what I'm trying to say, but then you have this person sitting next to you who can't sit still, or you have your phone that's buzzing in your pocket, or you have this heating and air vent above you making strange sounds, or you hear somebody outside in the hallway walking by and laughing. What keeps your head focused on me? It's because you can make decisions You're not responding to all stimuli. Can you imagine if that was the case? We would be so fidgety. We would be like looking at every, we wouldn't be able to focus. Kind of like with people with ADD or ADHD, isn't it? So people with those kind of situations may have to need some help to say, listen, let's focus. Let's turn down all of the external stimuli and let's focus. And there are some neurotransmitters that can help with that. Okay, local potentials. They accumulate at the trigger zone. So here's a picture of that. Maybe a better one than what I drew. You can see a bunch of arrows accumulating at the trigger zone, yay, yay, yay. All of these local potentials have to deal with mini sparks, little sparks. Now this one is really important. This is always on the test, typically, so I don't know how to put this on the test, but I'll try to figure it out. This is photo worthy. We're going to end with this slide, and we have a few more slides to go, but we'll pick up with this on Thursday. But let's end right here. There are some major terms. One of the terms that we talked about with muscles is a dotted line called threshold. We said threshold was this imaginary level that says that muscles had to achieve threshold to get a physical response. Below threshold, anything below threshold is what we call sub-threshold. And notice that the resting membrane potential of nerve cells is very, very negative. Just like it was in muscle cells, very, very negative. But if we get some local potentials, especially ones that are excitatory, that accumulate in the trigger zone, and it causes more sodium to be present, it raises, look, the charge to threshold. And once we get to this dotted line, oh my gosh, all of the sodium gates open, and we have this big spike where it says sodium rushing in. We go from -70 to +35. Depolarization, when sodium rushes in, sodium is positive. Then it drops, right? Because we have repolarization, potassium's kicked out. And notice down here that before it returns back to its normal homeostatic level, it dips below normal resting membrane potential, just temporarily. And we call this hyperpolarization. And underline that. When you drink excessively, you utilize a neurotransmitter that causes hyperpolarization, which means your nerves, listen, are less responsive than, or more, let's put it this way, your nerves are even more negative than it is at rest. So it takes even more effort to get a response. That's why you're slower to respond, you're slower to speak, you're slower to have reflexes because of hyperpolarization. That's got to be kind of interesting. So we know an action potential, right? It's when depolarization is followed by repolarization. Okay, so this is a money figure. So we drew some money diagrams. Take a picture of this, make sure you can explain this. I think you'll be in good shape. Okay, so we'll pick up from this slide, finish the last 4 or 5 slides, and move into the next one on Thursday. All right, I think lab is a little longer tonight. I think it's going to probably be like an hour and 20 minutes. So we'll start at 7 o'clock.